Depending on the routes they choose, some populations may arrive late to their breeding grounds, which can act as a pre-zygotic barrier. Here are some examples of different migratory routes taken by different populations to reach their overwintering sites. So these migratory divide will considered as behavioral barriers to gene flow. Apart from behavioral barriers, there could be historical barriers like last glacial maximum, or there can be physical barriers like Rocky Mountains in the west. So what happens when there is no barriers? There is gene flow, so population can mix with each other, but when in the presence of a physical barrier or behavioral barrier or historical barrier, gene flow is restricted, so the population can lead to genetic differences. In my study species, barn swallows, Dr. Hobson and their team found out there is an population migratory divide between western and eastern populations of barn swallows using isotopes analysis. Also, other research groups found out there is migratory difference in different populations of bank swallows. This is a snapshot of Motus wildlife tracking data. So that lead to our research objectives. Are there population genetic differences across the migratory divide? If there is genetic differences, we want to understand the potential influence on gene flow and also provide insights into the conservation prioritization. To achieve our objectives, what we did, we collected samples, blood and feather samples from different banding stations and with help of different collaborators. And also we received tissue samples from museums, and what we did, we extracted DNA. We used different type of extraction methods depending on the analysis what we wanted to do, and we did microsatellite analysis, rat-seq, restriction associated digestive sequencing, and also low coverage whole genome sequencing. But for today's presentations, I will be only focusing on the results of low coverage whole genome sequencing. This is principal component analysis for barn swallows using low coverage whole genome sequencing data. So all circles represent breeding samples, triangles are migratory samples, and squares are overwintering samples. I also use different shades of red to represent west of the Rockies, purple for the central part, and then shades of blue for eastern populations. So what we found out with principal component 1 and 2, there is a loose cluster for the west of the Rockies and tight cluster for the east of the Rockies. And when I check principal component 1 versus principal component 4, I found out within the eastern cluster there is also a central cluster and a east cluster. Here's the principal component analysis for bank swallows. With principal component 1 and 2, we can also observe similar type of pattern west of the Rockies, central cluster and eastern cluster. However, for some populations, we have only one sample from that site. So we are working on collecting additional samples from those sites as well as new samples from the other sites to expand the coverage of our study. Jumping into the discussion, so our first research objective was to identify are there population genetic differences across the migratory. So for barn swallows, yes, there are population genetic differences across the migratory divide. In addition, we also found out there is another cluster within the western cluster. So that means correspond to west, central and eastern part of the North America. Considering into bank swallows, we also found out there is 3 main population clusters correspond to the west, central, and eastern North America. So our second objective was to understand the potential influence on gene flow. The most likely explanation to our research would be different populations were resided in different separate glacial refuges during the last ice age, shaping their migratory routes. Also, historical events and contemporary barriers like Rocky Mountains and rivers further influence their migratory paths, resulting in the genetic structure that we can observe today. The last objective was to provide insights into conservation prioritization. So, as we all know, eastern part of North America experienced rapid population decline compared to the other parts, and also we can see the tight genetic clusters within eastern population for both species, which lead to conservation prioritization of the eastern populations of both species. With that, I would like to thank everyone, especially all the present and past members of the Bird Lab, all the collaborators from bird banding stations, museums, and all collaborators who sent us samples, and also for funding Environment and Climate Change Canada, INSERC, and University of Lethbridge. And also, if you have any questions, please reach me. Thank you so much.
All right, excellent talk. We do have the author here in the audience. So if anyone I guess I should first check. Can we have our author unmute? Are you able to receive questions? Okay,
Yeah.
perfect. Thank you. All right, so at this time, anybody who has a question is able to raise their hand, and the author will call on you to ask your question. All right, so I had a question just to get things rolling. This was part of your master's thesis, and obviously this was a ton of work, really, really good stuff. So what's next for you?
So we are in the process of increasing our sampling coverage. So we trying to include more samples for bank swallows and both barn swallows. That's what I'm doing right now.
Excellent. And you have a question in the chat.
Oh, not that I think of. We didn't find any evidence for hybridization though.
And you have a second question. I'll read it so everyone can hear. Great talk related to Daniel's question. How have population ranges shifted, if at all, with climate change?
Yeah, so we haven't looked into that, but we were also thinking about checking the ecological niche modeling and considering environmental factors to see if there is an environmental factors leading to these genetic differences and all. So that's also would be the next part.
Right. So are there any further questions for our speaker? One more. It looked like maybe one population from the central group was clustering with the West. Any ideas as to why?
You mean for the barn swallow? There is a one triangle which is a migratory samples. So there could be is that what you're referring to? Yeah. So yeah, so that is a migratory sample. So we aren't sure that could be the maybe the western up. So we collected that sample while the bird is migrating. So that could be a western that could be a western population sample or central population sample. So it is a migratory sample, if that makes sense.
So it looks like our question answer was happy with that answer.
Mm.
So at this point, we'll have you go back on mute and we'll start queuing up the slides for our second talk, which will begin at 10:15. Right, so Denise, I see you there with your camera on. Would you like to try sharing your slides?
Yes, for sure. Let me do that. Yes.
Beautiful.
Are you able to see the slides okay?
Yes, that's
Okay,
perfect.
awesome.
You have there.
Right there.
And I'll introduce you in just a couple of
Yeah, it's okay. Thank you.
All right, and for our second talk in this session, we will be hearing about sexual dimorphism in orchid bees. All right, take it away, Denise.
Thank you very much. Hi, everyone. Thank you for those joining this session today. I'm very excited to talk about my work about exploring sexual dimorphism in an orchid bee, specifically Euglossa dilemma. I'm a current PhD candidate at the University of California, Davis, associated with the Department of Evolution and Ecology. So before we start, I want us to start thinking about the vast majority of the diversity of extant animals on Earth. And that diversity is mainly covered by insects, having more than 60% of the total diversity of current animals. And this diversity has been accompanied with a wide range of behavioral and ecological traits. So for example, we have dung beetles that are able to navigate deserts using the light from the moon for the sky compass orientation, or we also have migratory monarch butterflies that have a magnetic compass to know the direction of their migration, or widely recognized economically important pollinators such as honeybees that also form very complex social societies. And this diversity, such like invertebrates, can be traced back to the brain. So all of these behaviors are controlled by the brain. And what we're seeing here is just a representation of a mammalian and a reptilian brain with different color codes, which represent different brain regions that we know are associated with either a sensory modality or a cognitive process. And analogous to these brains, we have the brains of arthropods, such as an insect brain or a decapod brain, which also has different compartments that are specialized for different functions. And is this separation of compartments that allows us to understand and explore how behavior and brain are correlated? Because we know that modifications in brain structures actually can inform sites of adaptive behaviors by the differential expansion of different brain regions. So this is just an example. Here we have the waggle dance in honeybees. So for those that may be familiarized with this, they already know the story. For those that don't, honeybees have a form of communicating foraging patches with their conspecifics in the colony. And the way they do this is by basically doing a dance that is very visual. And what we see in the brain is that they have enlargement in their primary visual centering processes. So the medulla and the lobula are very large in honeybees. And again, these are visual centers. And on the contrary, for example, we have ants that are mainly subterranean and live in dark habitats. And we see an extreme reduction in these brain regions. And on the contrary, we see an expansion, a relative expansion of the antennal lobe, which is an olfactory brain region, is the primary center for olfactory communication. And so this leads me to my study system, which is the orchid bee glossal dilemma. And I'm very interested in study this species because I think it provides us a powerful system to understand how different brain components can inform on the sides of complex behaviors. And this is because we have a strong behavioral dichotomy in this species. So females are what we call facultatively social. So they create small cooperative colonies where we're going to have one dominant bee that is just going to stay in the nest and is going to be like the mother of the subordinate bees, which are going to be the ones that are going to be foraging and looking for both nesting materials and food sources to provide for their brood cells. And then on the contrary, we have solitary males and orchid bee males, contrary to many other species, they don't synthesize their own pheromones to attract females, but instead they rely on chemical compounds they collect from their environment to concoct a species-specific perfume. And this perfume is then stored in the hind leg pocket, that is the one you're seeing here in gray. And then when the perfume is ready, they will display that perfume, and the female is the one that actually is going to seek the male if she's attracted to that perfume. And also males, contrary to females, have wider foraging ranges, and we also know that they forage later at night during dim light conditions of light. And so I wanted to understand basically if there is sexual dimorphism in different brain structures at the intraspecific level. And so the way I did this was using micro-CT scanning and reconstruction analysis. So what you're seeing here is basically just the raw scans of the bee brain. I used an iodine contrast to increase neural tissue enhancement. This is just the segmentation process. I used 12 female bees and 12 male bees. They were all newly emerged bees to control for any effect of either age or experience in the brain. And then I reconstructed the standardized brain atlas for both males and females. So in the upper panel, you see the standard atlas of the females. And then in the lower panel, you see the standard atlas of the males, color-coded with the different brain regions of interest. So after I done this, I was able basically to calculate either differences in allometric scaling or relative investment using major standardized major axis regressions. So don't really worry about the formula here, but basically our region of interest is going to be a function of both the alpha, which is basically the relative investment, which is given by the intercept of the regression, the beta, that is basically the slope, which is going to tell us the allometric scaling of that brain region compared to the allometric control, which in this case was the remaining central brain. So the first case that we have, again, would be a change in slope, meaning that either males or females are scaling a specific area of the brain differently to the other sex. We can also have changes in the relative investment, which means that the regressions fall into the same category, but one is investing more by having a higher intercept in that regression line, or we can just have shifts along the shared slope, which means that they're relatively investing the same way. Just as things get bigger, all the brain gets bigger. And so when we ran this analysis, the first thing that I find is that males invest more in all primary optic regions compared to females. So primary optic centers are not involved in cognitive processes, but are just involved on sensory processing. So the first region that you see enlarged in males compared to females is what we call the lamina. So here is the regression lines for males and for females. Again, what we're seeing here is case number 2, where we have a higher intercept in males compared to females. And the lamina is known to play an important role in early color processing, sky compass navigation, which is basically used by polarized light of the sun, and contour determination by light and dark shadow contrast. The second region is the medulla, which is the yellow region coded here. And this is the region where the first full chromatic representation of the visual environment is performed in the bee brain. It's also involved in the sky compass orientation, direction sensitivity, and polarized light. And finally, we have an enlargement in the lobula, which is involved in motion detection and shape. So all of these primary centers of visual sensory processing are being enlarged in males compared to females. And this could be potentially attributed to the fact that males, we know that they forage later at night, and these conditions require higher demands on the visual system compared to when we have more light available for the bees. So to explore this a little bit farther, I performed a scanning electron microscopy on the compound eyes of both males and females to get into the details of some optical parameters. So I looked at the average facets per 12 squares in the compound eye to get an estimate of the facet density in the eye, as well as the average maximum facet diameter. And these two parameters are important because we know that typically changes in these parameters are either associated with an increased sensitivity or increased resolution. And these tend to create a geometric trade-off in visual systems. So when we have a larger diameter, or a larger facet diameter, we're increasing sensitivity because we're enlarging the surface area that is able to capture light from the environment. But on the contrary, by doing that, we're also decreasing the density of the facets that are together. And by doing that, we're increasing what we call the intermaterial angle distance, which as soon as when you increase that, you have less lower acuity. So when you have larger facet density, you have higher acuity because you decrease that intermaterial angle. So what we're seeing in euglossa dilemma is that males are having wider facets, increasing sensitivity. And again, these might be related to the fact that they forage at dim light. So they need to basically be able to extract whatever light is available in their environment, while females are actually packing their facets more densely by decreasing the length of their average facet, and this is able to increase spatial resolution. The other part of the brain that shows strong sexual dimorphism is what we know as the mushroom body, which is a higher order processing center for learning and memory. And many authors have even described this as the soul of the insect. All cognitive processes are determined in the mushroom body and is the main area of the brain that controls complex behaviors in insects. And what we see here is that in this case, females are the ones that are relatively investing more in this processing center compared to males, seen here again by the regression lines where females have a higher intercept compared to the males. Here is the three-dimensional reconstruction of the mushroom body of the females and the mushroom body of the males. And additionally, like what I did was I segmented the cluster of Kenyan cells, which are the neurons associated with this processing center, to estimate if we also had a larger number of neurons associated with the structure, because mathematical modeling has shown that increased numbers of Kenyan cells are associated with increased memory capacity. And this is actually exactly what we see. Females also have a significant larger number of Kenyan cells compared to males, and that actually correlates as well with an increased surface area of the calyx that is basically this cup-like structure that we see in the mushroom body. So this is interesting because it's actually kind of analogous to what happens with the neocortex. As you increase the foldings in the neocortex, you're increasing that surface area, and that gives you the possibility to play with the volume to surface area ratio, and so also the capacity to hold more neurons in your brain. And another important and interesting factor of this brain region is that in Hymenoptera, which means bees, wasps, and ants, this brain region is subdivided into three different compartments known as the lip, the collar, and the basal ring. And I just want you to know here in this case that the lip is center only receiving information from the antennal lobes, which means olfactory information. And then the collar is only receiving information from the visual systems. So it's just for vision information. And what we're seeing here is that the lip actually is driving that volumetric expansion in the mushroom bodies compared to the males. So olfaction is playing a key role in the volumetric expansion of the mushroom bodies when we compare the sexes. And these could be potentially related to the fact that social communication, again, is mainly mediated by chemical signals. And so this is just a video of how orchid bees look in the wild. We have the dominant bee here, which is constantly antenating the brood cells, making sure that the eggs are developing correctly. And here we have the subordinate, which is also in constant communication with the dominant bee, which determines the behaviors of foraging and nesting. And lastly, just to finish the talk, I want to say that after we looked at the different brain regions, we however see that both sexes share the same body to brain allometry, which means that it's more the allocation of the energy investment that is really driving those volumetric differences rather than a complete investment on neural tissue. So both sexes are actually investing the same as a complete, like as a full net amount of energy investment in brain regions, but they're allocating those resources differently, where males are allocating more in the visual system. And again, this could be related to the fact that they forage later at night and they have larger foraging ranges, while females are local foragers. So females don't need to remember as much landmarks as males do because they forage for longer distances. And females invest more in olfactory processing centers, and this could be attributed as a potential adaptation for social communication. And as I have the time, I'm just going to show you a quick video of the reconstructed brain of Uglosa dilemma and the advantages of using micro-CT scanning as a non-destructive method for the visualization of soft, non-mineralized tissue. So all of the brain regions that you see here, I tested for sexual dimorphism. Many of them didn't show any difference between males and females. The only ones that did were the ones that I mentioned through the talk. There are other differences that I'm happy to talk or discuss if you have any questions. And with that, I'd like to thank you all, and I receive any questions that you may have.
All right, excellent talk. We're a little short on time, so I'm going to have the next speaker get their slides up. But we do have time for a quick question if anybody would like to ask one. All right, so we have a question in the chat. I'll read it out so that you can hear it. Do you plan to test for behavioral differences between males and females for sensitivity in dim light versus spatial resolution?
With their environment. Yeah, I think that's a great question. I am planning, well, after I did the standard brain atlas, I'm currently looking at plasticity in different brain regions to see if there are certain brain regions that are changing through experience or age of the bees. So I think I might do a trial kind of testing how well they perform in different light conditions. I really haven't thought about doing that, but thank you actually for that idea. I think it's a great thing to test.
Okay, excellent. And that's about all the time we have here. And we have
It's okay.
Yeah, thank you. And we've got to move on to our final talk. So Manuel, are you ready?
Yeah, hello.
All right, take it away.
So, hello everyone. My name is Manolis Meranveliotakis. Thank you for being here. My talk is more methodological. It's about a pipeline that we are building to mine universal single copy orthologs from whole genome short read sequencing data. So, when we talk about universal single copy orthologs or USCOs, we talk about coding loci that they are present as a single copy within the genomes of the taxa that we are interested in. And since they are orthologs, of course, they originate from a common ancestor and they are separated by speciation. Now, these attributes, along with other characteristics that we can discuss afterwards if anyone is interested in, make USCOs very interesting set of loci for research in phylogenomics and systematics, and especially for species delimitation approaches, as they can provide a standardized set of markers for species delimitation across the tree of life that can go beyond the single locus approaches, like, for example, the CO1 barcode in animals. Now, the first use of USCOS was mostly as a benchmarking method in order to see how complete a whole genome assembly was through the Busco pipeline. But this has eventually been evolved into a more complete suite of tools that can do gene prediction and generate phylogenomic datasets. However, the Busco pipeline is heavily relied on the Ortho database, the OrthoDB, and the Busco datasets that exists within this database, which if you work on higher taxonomic ranks, it's perfect. But if you want to work on more shallow levels, there is a possibility that a group for your organism of interest is not yet included. And another potentially problematic part is that it needs whole genome assemblies, which if you only have short read sequencing data, it's not always so straightforward to accomplish. So for the latter part, the whole genome assembly, different approaches um, have been published using reduced representation sequencing like target enrichment, where they created probes in order to get these Busco loci from different genomes without a whole genome assembly. And these are actually brilliant approaches, but again, you have to have the prior information that exists in the Busco, in the OrthoDB database in order to generate the physical probes and actually get your loci. So here I'm going to focus on a different approach. It's called local assembly approach. It doesn't need a whole genome assembly, just a set of short read data from shotgun sequencing, and it uses baits, in silico baits, meaning that there are no physical probes. Just to put it simply, some sequences that you are interested in and you can find in several public databases like NCBI. And what the local assembly does, well, it's in the name. It takes each of these sequences that you have as baits and tries to assemble them for the genome that you have sequenced. So imagine that you are interested in a non-model organism, let's say a genus of beetles that let's say it's taxonomically placed in the family Tenebrionidae. So what you have to do first is to create your in silico bait set, and we wanted to use something specific for Coleoptera, but this did not exist in the Busco datasets. So what we did was we downloaded any genome with good assembly, good set of genes available in NCBI, and we used OrthoFinder in order to generate thousands of universal single copy orthologs, either for the whole order of Coleoptera or for specifically for the Tenebrionidae family that we were interested in. And based on some filters that we applied on the length of these baits, we kept around 5,000 to use as these in silico baits. So the next step is to use a software that actually does this local assembly approach. We chose to use the SRA assembler, which is a very nice software, very modular. And what you get from after running your shotgun, your sequencing, your short reads from shortcut sequencing and your baits, you get either a very nice complete assembly, meaning that all your exons from the bait of interest are within the same contig, or you get a fragmented assembly where you can find exons, but they are in different contigs. So this was the first problem that we had to solve, because even though we could see that there were interesting information in these fragmented assemblies. SRA assembler did not consider them and only considered the complete assemblies. So we created a script that actually uses the information from complete assemblies of one specimen, specifically the coordinates that this assembly aligns with the initial bait in order to get the exons from another specimen and actually check them, check their quality based on some similarity indices that we implemented, and also trim any possible erroneous regions of this exon. And then take everything, stitch it together, and create this your phylogenomic, let's say, dataset for your specimens. So to visualize that, imagine that you have two specimens. From the first one, you get the blue locus, locus 1. From the second, the green locus, number 3, and you lose everything else. After the rescue approach, you could use the information from one sample to actually get more loci across all your specimens. So we checked how many loci we were getting with each method for different sequencing coverage, 10x and 20x. And we saw, you can see with blue what you were getting with SRA assembler. This is the number of specimens that the locus which is assembled is present. And this is the number of loci for each category. So with blue, you can see what we were getting, and with red, what we got, and with red and orange, what we got after the rescue approach. And we almost got, well, in one case, we got 100% of 1,000 randomly selected baits across all 19 specimens. And with 10x, we got almost all, we got 97%. However, the question here is, what is it that you are assembling? Because we couldn't actually check for errors implemented by the assembler or sites for low coverage because this information were not available. So what we did was we took for each specimen the raw reads and we remapped them on the assembled baits in order to get this information. And then we could actually remove any regions of low coverage and identify erroneous reads that the assembler had selected instead of other available reads that were more fitting for the assembly. So after following these steps and also removing sequences with more than 25% of missing data based on the locus length, you can see that even for a very strict threshold of 90% specimen coverage, you get almost 3,000 loci for downstream analysis. And even for a more strict 100% specimen coverage threshold, you get more than 1,000 loci. So at this point, you have a very nice set of thousands and thousands of loci for many samples. And the question is, how do you check thousands of alignments, at least in an automated manner? So we started checking the problems that they were present in our alignments. The first thing that we found was that there were alignments with in-frame stop codons, but these in-frame stop codons were always located in areas such as this one. And this is probably because an intronic region has been incorporated between the different exons, because in the organism that we took the baits from as our reference, since it's not a very closely related species, just the only available related species, this area might be an exon, so it has been assembled for our organisms as well. So we were conservative and we thought that we should remove any of these alignments as we couldn't really be certain for its validity. The fourth problem, which is related to the one before, is out-of-frame blocks. Again, this type of problem is probably due to an intron being incorporated. So again, we remove such alignments. And the final step was the fact that we saw the presence of artificial orthologs. You can clearly see there are two very distinct blocks here. I probably forgot to tell you. So, for example, the organism that was of interest to us was a genus of different beetle species, and you don't expect such variation within this genus, so many substitutions. So what we did was to get a distribution of how many variable sites exist per alignment and identify those outlier alignments and also remove them. And we saw that this indeed removed any possible artificial orthologs from the dataset. And after implementing all these filters, you remove more or less a 10% of the previous amount of loci. So still for a 90% threshold coverage, you get a little more than 2,600 loci. And for 100% specimen coverage, you get even more than 1,000 loci ready for downstream analysis. And also, if anyone is interested in the Busco datasets, you can find also these Busco sequences within the assembled datasets. For example, for the metazoa initially we included 650 as baits, and we mined more than 400 of them. And that's it for the pipeline. This is just let's say a proof of concept. This is a phylogeny that was created. The pipeline is still in progress. We are planning to include the mining of introns as well. But for example, here is this genus of beetles that I was talking about. With black, you can see between species branches. With orange, you can see within species branches. And you can see that most of the between species relationships, if not all, are very well resolved. And I think that my time is up. So I will finish here and let you ask me any questions that you might have. So thank you. That's all.
Excellent talk. And so this actually does conclude our session. Our fourth talk did withdraw. And so we can, people are welcome to leave. So if you did arrive, I saw a few people arrive in the last minute of that talk, but unfortunately our fourth speaker for this session did withdraw. So that fourth talk is not happening. And so if anybody has a question that they would like to ask our speaker, you are certainly welcome to do that now, since this is the end of the session. Feel free to raise your hand if you do have a question. We can take one or two. All right, well, that brings us to the end of our session. So for timing's sake, we will end here, and we have, our speakers are available in the lobby if you have additional questions, or I believe you are also in our directory.